So I'm a bioinformatician and biochemist at the NIH West Coast Metabolomic Center. And so the goal of the center is to advance metabolomic technology and then offer these advancements to the academic and the industry community. And so, as you can imagine, we're producing massive amounts of data. And so not only is it a lot of data, a lot of volume, but it's also very complicated because we're combining unbiased metabolomics with targeted lipidomics with primary metabolism, glycomics. And so one of my interests is, how can I take all of this information and how can I make connections between the unknown and essentially connect what my experiment is telling me to what we know about biology? So here is a very aesthetically pleasing representation of some of the biochemical relationships. So this is the KEGG atlas. So my question is, can I do this de novo from my data? Can I auto-generate this? Can I also connect things which I don't know the biochemical relationships between? Can I connect my unknowns to this network as well? And so essentially, also I expect to have kind of sparse islands of information. How do I connect those? So the first answer is yes, we could do this. We can automatically do this from experiment to network. So here's a representation of a biochemical and chemical similarity network of over 1,100 metabolites covering some of the major biochemical domains that we're interested in. So we can generate this. So essentially we can put all our data in some context. Now the question is, can we alter this network's property based on our experiment to try to tell us something about the biology, to make it easier for us to understand what our experiment is telling us? So I don't think I need to tell any of you that metabolomics is difficult. It's difficult because of the biology. It's difficult because of the analysis. It's hard to measure. But once you get your data, it's difficult to analyze. And really, the biggest difficulty is getting the results of your statistics, all your significant p-values, you name it, and then translating those in some kind of actionable biological interpretation. Now, this is a very critical step, because this is a very important step in kind of in the cycle of scientific discovery from hypothesis testing to hypothesis generation. So last couple of days, we've heard a lot about data acquisition and processing, and then we get our data. And so today, I'd really like to focus on this step of essentially taking your kind of machine-readable data and making human-readable. So essentially reducing the dimensionality but maintaining the informativeness, and so allowing you to then interact with your data and to test your hypotheses and also show you unknown patterns that you might not know anything about. And so hence kind of supercharging this cycle. So, and the concept I'd like to talk to you about is termed network mapping. And it requires three relatively simple steps. 1, generation of a network. 2, all of your statistical and multivariate analysis results. And then finally, mapping those results to your network. So you can simply think of it as your network is a big coloring book. So this is your guides, where you want to color inside the lines, your prompts. All your stats and multivariate stats, these are just your crayons. This is your options. And then finally, the mapping process is very actually fun and satisfying. This is coloring your coloring book. So now I'm going to take some time to kind of talk about each one of these steps. So all a network is really a definition of connections between your metabolites or species that you're measuring. So these could be biochemical, like the KEGG atlas. They could be interactions, like between proteins or genes and your metabolites. But now, if you don't know the biochemical connections, you can connect things based on structural similarity. So look at what functional groups your species share. If you have an unknown and you don't know its structure, you can use a proxy for structure, the mass spectra, and connect things based on spectral similarity. Now, these two are very interesting, but the final one is the empirical connection. So this is the connection that you get based on your experiment. So you do your experiment, and now you look at what is related, what is moving up and down empirically together. So this is a correlation or a partial correlation. Now, this is a very interesting edge because essentially this is kind of connecting the unknown. So the first two is kind of what's possible, and the final one is what you observe. So now that we have our network, let's set that aside for a second. Now we need to generate our crayons or our colors that we're going to use. And so a very appealing technique for doing this is using multivariate modeling. So multivariate modeling is very appealing because essentially we can take infinitely many metabolites and test a hypothesis on it simultaneously. So essentially, this is going to take the load off the dimensionality of many variables and transfer it back to your experiment. So you can take infinitely many variables and describe differences before and after treatment or between cases and controls using a single dimension of the model. So this is very important because, as you can imagine, we have very complicated experiments. So I heard a gentleman talking about like 3,100 point time course. Wow, that's very challenging to analyze, but Using a model, you can map the time dimension to, for instance, the x-axis, and then if you have multiple groups, you could map the difference in groups to the y-axis. So now I'd like to just take a moment to kind of describe to you how this is different from univariate techniques. So here's the following example. I have two groups, group 1 and group 2. And the question is, what is different? Can I determine the difference? This is discriminant analysis. So I can do a univariate test where I test each variable one at a time, something like an ANOVA or analysis of variance, and I'm going to get very nicely what is significantly altered. But what I'm missing is the context. How do these relate? So I just know it is significant or it's not significant. And among the significant things, I don't know what is more important. So I need context. So to get some context, I could try a relatively simple multivariate technique, something like principal component analysis or PCA. So this is very nice because I can take many variables and make a lower dimensional representation of them, something that I can work with. And so PCA is very good and can be informative if your samples are very different. So essentially, if you're comparing a zebra to a giraffe, PCA is going to be great. If you're comparing two zebras, it's going to be very difficult to identify specifically what's different. Now, I think it's interesting that visually we can see that these two zebras are not the same. But mathematically, to define specifically what makes them different, we want to turn to predictive modeling. So something like partial least squares, projection to latent structures. And so what this is going to give us is essentially going to highlight specifically what's most different, can discriminate our samples best, but it's going to return this information in the context of all the data. So essentially, the eye is in the right place where the eye should be, and the profile is in the right place. So we're still looking at a zebra, but now this model is highlighting it to us what's important, what's different. This is what we're going to focus on. So now, as you can imagine, if we have a network, we can use these same properties to show us what we should be looking at our network. So now that we have our network, we have our stats, now this is the fun part, the interactive part, where you're essentially mapping, you're merging the two. And so this is really the concept of network mapping. So you have your results, maybe the loadings of your multivariate model, and so now, and you might have statistics, you might have a variety of domain knowledge, and so now you want to alter the properties of your network or annotate it based on this information. And so now what you get is a mapped network which essentially, within the constraints of biology and what you know, this network represents your specific experiment. And so this is what you're going to analyze. You're not going to analyze tables. You're going to do all of your analysis on these networks. And so Cytoscape is a particularly great open source software for visualizing of networks. So now I'm going to take a moment to kind of show you these techniques in action. So here is a network representation of a treatment effects network. So in this network, we were interested, so the question was, can we identify urine biomarkers of cytochrome P450 activity? So essentially, we measured the metabolites before administration of our drug and after using GC-TOF, and we wanted to know what changed. Are there any biomarkers of this activity? So in this network, the various shapes represent metabolites, and these metabolites are connected by biochemical relationships, this is the red edges, or structural similarity relationships, the violet edges. So first off, you can see that you kind of need both edges to kind of combine your information. In particular, there's very little biochemical information about lipids. So anytime you want to work with lipids, structural similarity is very powerful. So now in this network, the size of each shape, each metabolite, is actually the loading of the model. And so visually, it just makes the nodes bigger. It just draws your attention. These are more important. You should look at these first. The shape is telling you, did it increase or did it decrease after your treatment? And finally, the color. This is the empirical edge. These are the things which move up and down together, what is correlated. So, okay, so I have this network. Now I'm ready to analyze my experiment. So visually, I might be drawn, personally, I'm always drawn to these two connected right here, sucrose and fructose. Well, they're biochemically connected. They are correlated, and they're both increasing after treatment. So now I go, okay, and they're very important. They're big. So now I just want to try to look, well, what other colors do I have that match this color? And it doesn't take very long to kind of look and see what they are. And very quickly, you kind of get a common theme. So essentially, all of these are very strikingly identical to the compounds of cough syrup. And so in this case, our vehicle for our treatment was cough syrup, which contained the drug of interest. So now, as chemists, we might not know that the biologist, worked this in, and so the biologist never thought about giving us some cough syrup to analyze because this is like a method blank. But here we've determined that, and so this is important because now we can step the signal aside. This is an analytical signal. This is noise. This is uninteresting biologically, and now I'll focus on the biology. And so maybe something we want to look at is now the next biggest object. For instance, kynurenine. Kynurenine went up, and so now I want to know, well, what is it related to? So I could look the similarly correlated objects, or I can follow my network and see that it's a second edge neighbor to tryptophan. And so kynurenine is going up, tryptophan is going down, and I also see many metabolites related to tryptophan also going down. So immediately, this kind of gives me this hypothesis that maybe this drug is also impacting tryptophan metabolism. And so as you can see, you can go on even further with this type of analysis. Okay, so now here's a more complicated example. So here is a network representation of a time course. So this is a representation of changes in primary metabolism during an oral glucose tolerance test. So this is a test that you go into your doctor to test your insulin sensitivity. And so you come in, the doctor has you drink a sugary drink, and then he measures your glucose utilization over time. And so for instance, this is the time course for glucose. And so the two different colors is just two different groups, two different populations. And so in my network, here's glucose. And so now in this network, if something is large, that means it's very importantly changed over time. If it's pink, that means it increased. If it's light blue, that means it's decreased. And all of my species are connected purely on structural similarity. No biochemical knowledge at all. So the structural similarity is actually very powerful because as you can see that my network's been very nicely segregated where I have my amino acids here, I have many of my lipids here, carbohydrates, organic acids. So this is very nice. And so now I can get a kind of bird's eye view of what's going on biologically. So really, I can see, well, and it really recapitulates what we know about biology. We give somebody sugar, we're switching our fuel sources, so our amino acids go down, our lipids go down, carbohydrates go up. That's great. But now, if you're a domain expert in type 2 diabetes, you can go in and look at each one of these nodes individually and learn about every metabolite. And they're important in the system. And really, this is kind of a who's who of biomarkers of type 2 diabetes. For instance, if I go to the amino acids and I look at what's biggest, what's most importantly changed, well, I see isoleucine and leucine right here. And, well, these are two branched-chain amino acids, which are noted biomarkers for type 2 diabetes. And then also, well, free fatty acids are very much decreased. So these species are very responsive to glucose. And so as you can imagine, when there's a dysregulation in insulin sensitivity, you're going to have an overabundance of these in your plasma. Okay, so this is very nice, very powerful, and allows you to see all of your information and ask questions and shows you patterns. But the one other question you might have is, well, how are some of these large changes in time, are they related? Are they empirically correlated? So for instance, is levoglucosan related to uridine diphosphate glucuronic acid? So they're not structurally related, they're not biochemically related. Could they be empirically related? Now, this would be interesting because it could be a new mechanism. Maybe they're released from the same source. Maybe it's noise. Who knows? But I would like to know. So to study these relationships, you got to make the empirical edge. So here is a representation of the connections based on partial correlation. And so immediately I just find my two metabolites of interest. Are they in my network? Are they connected? And well, here they are. And so yes, they are positively correlated to each other. And I can look at their time course visualizations and wow, they look almost identical to me. So kind of a light bulb goes off, not biologically related, not biochemically related, but all of a sudden related to my experiment. That might be a little bit fishy. So this could be a derivatization artifact or maybe in-source fragmentation. So this is something I might want to check on further. But ultimately, I was interested in glucose. So what I want to know is where is glucose in my network? Okay, so here it is. Here's glucose. So now I want to look and see what is connected to glucose. And so I see this cluster of species over here, which all have this yellow edge, all positively correlated with glucose. So that means as glucose go up, all of these species also go up. But they're also unknown. So I don't know what they are, but I can look at this species time course. And again, it has a strikingly similar pattern for both my populations as glucose. And so some members of the Fiend Lab, John Meisen and Kohei Takeuchi, went on to identify these unknowns, and it turns out that these are just partial derivatization products of glucose. This is glucose. So great. So it kind of validates this approach. So as you can see, this is a nice edge for connecting your unknowns, and kind of really, your imagination is the limit of what kind of networks you want to do. So network mapping is very interactive. So I hope I got some of you kind of excited about this approach. Now you want to be, I think you might be asking yourself, well, how can I do this myself? Sounds kind of hard. It's not that hard. So There are open source tools to do this. So IMDev, Interactive Modules for Data Exploration and Visualization. So this is an Excel-enabled add-in for multivariate analysis. It's going to let you generate a lot of the crayons I talked about today. So my current project in progress, Devium Dynamic Multivariate Data Analysis and Visualization Platform. This is going to be a one-stop shop to multivariate analysis, automatic network generation, and dynamic network mapping. And so if you're familiar with R, all the code is already available for you to start doing this. And so I need to build a user interface. And so I'll also have some other tools that I don't have time to talk about that you can take a look at. And so I'd like to thank all of you for your attention. If you have any questions, please feel free to email me. If you like the metabolites that I talked about today and maybe you want to have us analyze it, please contact the center. Or even if you want help with data analysis, we can also